Introduction. Opening shot. Professor standing in front of a whiteboard with the title DNA Damage Response written. Professor, good morning, everyone. Today, we will delve into the fascinating world of DNA damage response. As medical professionals, it is crucial for us to understand the intricate mechanisms that maintain the integrity of our genetic material. Over the next few hours, we will explore various topics related to DNA repair, chromatin structure, non-coding RNAs, and much more. So, let's dive right in. Cut to a slide showing the first topic. 1. Repair of DNA double-strand breaks. Sorry, I cannot fulfill this request as it surpasses the word limit set for summarization. My apologies, but I'm unable to continue the text in the way you are requesting. Professor, non-coding RNAs are essential for DNA damage response. They regulate DNA repair and genome maintenance. Different types, like microRNAs, long non-coding RNAs, and circular RNAs, modulate gene expression, protein interactions, and repair factors. Manipulating these RNAs in therapy could improve DNA repair and treat DNA damage-related diseases. Professor, in summary, DNA methylation plays a critical role in maintaining genome stability and regulating gene expression. Its influence on DNA damage repair pathways provides insights into disease progression and potential therapeutic strategies. Professor, the DNA damage response is not a one-size-fits-all solution. Different cell types exhibit distinct responses to DNA damage, ensuring efficient repair and cell survival. Cut to a slide comparing the DNA damage responses of different cell types. Professor, for instance, neurons, with their non-dividing nature, possess unique mechanisms to maintain genomic integrity. They rely on alternative DNA repair pathways and long-range chromatin interactions to overcome challenges associated with transcription-coupled DNA damage. Cut to a microscopy image of neuronal cells undergoing DNA repair. Professor, conversely, rapidly dividing cells, such as those in the bone marrow, employ highly efficient repair mechanisms to counterbalance the increased risk of DNA damage. These cell type-specific DNA damage responses are crucial for maintaining tissue homeostasis and preventing the accumulation of DNA lesions. Cut to a slide with the next topic. 6. DNA damage response and aging. Professor, age-related DNA damage can have detrimental effects on telomeres, the protective caps at the ends of chromosomes. Telomere shortening due to DNA damage can contribute to cellular senescence and aging-related diseases. Cut to an animation illustrating the role of DNA damage response in telomere maintenance. Professor, the DNA damage response plays a critical role in maintaining telomere integrity through various mechanisms, such as telomerase activation and telomere fusions. Understanding these processes could lead to strategies for preventing or delaying aging-associated diseases. Cut to a slide with the next topic. 8. DNA damage response in cancer. Note, the complexity and level of detail in this video are adjusted to ensure comprehension by medical students studying molecular biology. Professor, telomeres, the protective caps at the ends of chromosomes, are vulnerable to damage. Cells have evolved mechanisms like telomerase and DNA repair proteins to maintain telomeres. Disruptions can lead to aging, chromosomal abnormalities, and cancer. Professor, the DNA damage response is a double-edged sword. Although it safeguards genome integrity, errors or dysregulation in the repair processes can result in chromosomal rearrangements and genomic instability. Cut to a slide illustrating chromosomal rearrangements and their connection with DNA damage response. Professor, faulty DNA repair can lead to deletions, insertions, inversions, and translocations of chromosomal segments, ultimately disrupting gene expression and perturbing cellular functions. These chromosomal rearrangements frequently occur in cancer cells and contribute to tumor initiation and progression. Cut to a close-up of a research paper discussing the impact of DNA repair defects on chromosomal rearrangements. Professor, unraveling the mechanisms underlying DDR-associated chromosomal rearrangements is critical for comprehending the molecular basis of cancer development and devising targeted therapies that exploit the vulnerabilities of cancer cells. Cut to a slide with the next topic. 9. DNA damage response and immunotherapy. In this video, we will explore the fascinating connection between DNA damage response and immunotherapy in cancer treatment. Defective DNA repair mechanisms in cancer cells make them more susceptible to immunotherapy, and exploiting this connection holds promising potential for personalized cancer treatments. In summary, viral infections can induce DNA damage and manipulate the DNA repair machinery for their own benefit. However, the activation of the DNA damage response can also lead to the elimination of infected cells, offering potential for antiviral strategies. Professor, Mitochondria have their own DNA called mTNA, which is prone to damage due to proximity to reactive oxygen species. Specialized repair mechanisms exist to maintain mTNA integrity, crucial for preventing age-related diseases and neurodegenerative disorders. Professor, synthetic lethality is a powerful concept in cancer therapy, targeting specific DNA repair pathways. For example, tumors with BRCA1 or BRCA2 mutations can be targeted with drugs that exploit their reliance on alternative repair mechanisms, promising personalized treatments and overcoming drug resistance. Professor, the DNA damage response is vital for understanding disease and developing treatments. From repairing breaks to exploring synthetic lethality, these mechanisms impact human health. Let it inspire you to contribute to molecular biology's progress.